Swayam Prabha. Digital India. Educated India.
Thank you very much for your kind introduction. And again, a very good morning from Denmark. I hope you can hear me well and I see the slides there as well. Thank you also for the organizer and for Professor Raj and Srinivas and his team for the kind invitation here. It's a pleasure to have this opportunity to talk to you and exchange our views on how modeling and PAC in combination with AI can contribute to, let's say, the emerging era of biology for manufacturing in life sciences. So I said I'm happy to acknowledge our relatively large PAC team at DTU Chemical Engineering, which allows us to tackle and contribute to different aspects of, let's say, life science process industry, from experimentation to modeling, simulation, control, among others. My colleague Jens, Jacob, Chris, who is our also center leader, my young colleague Sayed, Ulrich, who's our CFD specialist, and myself. So in the coming, hopefully 30 plus minutes, I hope to touch upon the really the promising potential of biotechnology as a manufacturing platform for a variety of products, looking at the expanding interest in the investment in this area, and touch upon really the one particular aspect, which in our opinion underestimated, is the scale up of the process. And here linked to our thoughts how modeling and AI together may contribute to this challenge. And then zoom out and look at some of the grand challenges, if you will, in this area of, let's say, realizing this potential of biology for manufacturing. Right. So, I mean, since the completion of the Human Genome Project back in the 2000s, there has been advances in decades in making in the area of sequencing, let's say, technologies, sequencing of DNA, largely known as omics. Also, the advancements in the gene editing capabilities, such as the CRISPR technology, and in addition to that, the advances in computing and AI, among other things, such as bioinformatics, has allowed the biological scientists to understand and increase their ability to really engineer the organisms, to discover many useful genes that instructs a variety of products, and then organize let's say, engineer the organisms to express this at the relevant titers and yields. And this has unleashed a number of euphoria and optimism to use the biology on its own as a manufacturing platform, moving beyond, let's say, manufacturing of penicillin or insulin or a number of other, let's say, therapeutic products, into more applications from food, from textile, like artificial silk, to chemicals and energy, among many other things, as is shown in this, let's say, a picture from the, let me see if I can highlight it here. Yeah, from the, let's say, position paper from the Bioeconomy Task Force from the DSA. Indeed, there's quite some excitement that for some, this century will be the area of the biology, akin to the previous century. 20th century was the area of chemistry or physics, depending on the viewpoint. And then, according to McKinsey report, is a promising economic potential expected from unlocking the potential of biology for product, manufacturing a variety of ends, anywhere between the $2 to $4 trillion in the coming 20 years. And McKinsey here foresee, impact into a number of areas in biomolecules, such as, for therapeutic applications, to biosystems, as in fermenting food, really, cultured meat, for example, to some other niche applications, such as biocomputing and biomachine interfaces. So there is quite some potential, economic potential there. And one thing I would really pause a second and touch here is the exciting development taking place, and in the area of using fermentation for the protein. Now, fermentation technology itself is as old as our civilization has old. We have used it to ferment, cheese, yogurt, wine, beer, you name it. And there we were working with the, wild strains, nature occurring and useful strains, right? And now, thanks to this advancement in engineering biology, and now the ambition is to discover genes useful to, let's say, responsible to make the proteins in various, let's say, food, like in the meat, to the protein in the eggs, or protein in the seafood, or various proteins in the human milk, or cow milk things. And once you do that, to try to express it in an engineered organisms, such that you can use the organisms to make it. So around this idea and the landscape, there has been a global fervor, if you will, a lot of initiative. So it is not just vision in the lab, in the academics lab, but it's really, there's a lot of, let's say, push to bring it to the reality. And this is just a map highlighting a number of startups and initiatives to bring this fermentation to produce really protein for food grade consumption. And according to one report, last year they have raised to the tune of $1.7 billion to allow to try and test these ideas. So just to pause, I'm showing here one example. It's from a company, Israeli startup, Remilk. They have raised also a significant amount of funding to realize their vision. So what they want is, they identify, using their synthetic biology platform, they identify a gene that is coding a protein in the cow milk. They have selected yeast as their host organism to try to express this gene with the instruction to produce this protein. And this is now you're replacing your cow basically with your fermentation. And once you produce this protein, which is present in the cow milk itself, then you use it as a kind of replacement to the cow milk, and to both to produce a number of dairy products. This is really the vision, and that vision, as we see, has been expanded to a number of other food proteins. And it is not just really, as I mentioned, as a kind of like a conceptual thought, an idea whether you can do that. There is a significant, let's say, investment which is being done, not only on the food proteins, but beyond, right? For example, this is news just from the 2020 that I could capture from my LinkedIn, if you will, newsfeed. Samsung Biologic expanded their contract manufacturing in their product. They are a contract manufacturing organization. In their campus in Korea, they doubled their capacity. Fujifilm, similarly, they invested in USA and also in Denmark here, quite large investment to double again their contract manufacturing facilities to allow to produce at scale. And Boehringer Ingelheim, similarly, they invested in Australia to increase their cell culture capabilities. AGC Biologics, similar, in 3 sites in Europe. Unilever partnered with Geno, a startup, to try to, what is Unilever, what's relevant for them, to try to produce a plant-based alternative, right? For example, the palm oil with renewable, we're using the bio-based manufacturing, yeah. Novo Nordisk, a company we know, we work together from Denmark. They have already actually started their expansion capacity, also to do quite kind of a big expansion facility here in Kalundborg to the tune of 17 billion Danish kroner, give and take around $2 billion thing. And Remilk, as I mentioned to you, they also started their greenfield project into establishing a precision fermentation facility to realize their vision. And there are many more. This is what can I capture. So is there sufficient capacity? Is there sufficient steel equipment to realize this potential? Maybe time will tell. Maybe this will lead to overcapacity or undercapacity. We'll really see. But having the equipment, the steel is one thing, right? What you need also, the recipe, the brains, right, to find out the conditions to establish the process. Because when you go from the discovery of the molecule from the lab to the at scale production, number of challenges rises to ensure the product quality and yields at larger scales. It's easier said than done, really. So you need to establish the process, including fermentation, the product capture, recovery, purification, among many other things. And often what we see in this excitement and euphoria of unlocking the biological potential as a manufacturing, the process is underestimated. There has been amazing, really, development in automation and high throughput screening and bioinformatics area to discover interesting molecules and engineer the host organism. But when you come to the, establishing the process in a reasonable time, because it takes time and resources, right? From the lab to the pilot to production scale, it's experimentally intensive. It is trial and error, and it is not always, given that you will find that process. So a case in point is Zymergen. It was the darling of it is maybe still, I don't know, the synthetic biology. So they have a very rich metagenomic database. They have used their synthetic biology platform. They screen for gene. They identify a product to produce hyalin in that case, which is a bio-based polymer that can be used for coatings in the electronic industry. So check, right? It's renewable, decarbonizing, raw materials for electronic industry. Wonderful, great excitement. It raised funding that valued the company at around $5 billion in 2021. But this year, this sold to Ginkgo Ventures at $300 million. So from $5 billion, your valuation dropped to $300 million in less than a year. And as soon it emerged, according to this, here you could follow up the details, that they have challenges with producing this promising product at scale due to the technical challenges with establishing the process together with their contract manufacturing partners to produce the product quality and high titer. And also they reported challenges with respect to the product testing and implementation in the electronic industry. So combine this, now basically this is a process scale of failure. This product was not able to be scaled in a relatively reasonable amount of time. And in fact, this is not the first time it's happening. If we go back a little bit to 2004, 2005, there was also quite some excitement about synthetic biology to manufacture a lot of synthetic fuels. At that time, we were faced with another crisis. The fossil fuel were very high, around $100 and $110 a barrel, and there were energy security concerns. So there were quite some try to use biorefinery to produce the synthetic biofuels there. And there were many startups at that time. And you can see this red colored here from my colleague, Gerger Stefanos from IIT there. You could see a number of startups that failed to realize the scale. So you can identify the gene, you can identify the interesting molecule, you can engineer your host organism, and that's great, but it is not given you're going to find that process that will allow you to produce it at commercial scale. Now, with this challenge, we believe the PSC community at large can help and contribute to facilitate, the process development, among other things, and help commercialize the potential of biology. And this picture is from 1983 about the nature of process system engineering, which is also reproduced from our colleagues' work, Wolfgang Marquardt and Claude from Aachen at the time. And the PSC has the ability to describe using modeling, right, fundamental phenomena, and to use it to, describe different unit operations, different equipment, fluidized bed, reactor distillation, fluid flow, and use this to try to design and evaluate a number of different processes from pulp and paper, fertilizer, refineries, chemicals, and this the community has demonstrated their ability to develop a number of skills and methods to help, right? And this hopefully should also help us to tackle or to take on the challenges that presents itself in biomanufacturing. And I'd like to pause here a second and highlight the importance of the models. I mean, at the end of the day, PSC is a dry lab, right? I mean, we are supposed to do wet lab. So in that sense, model, which in a generic form, usually it takes an input related to design decision, operational decision, take also constraints related to, let's say, external inputs related to feedstock composition variability, to commercial environment, right, when we want to evaluate the techno-economics. And then as an output, we'd like to have from our model, KPIs like the product quality, performance, operability, safety, among other things that hopefully will form the basis for maybe designing, scaling up, or operating a certain process. And in this regards, a model here, we have been working a lot with the ODE, PDE, state space type of models, but now thanks to the AI and machine learning, we can also work with, let's say, deep learning or other kind of models as well. So they are important. Once we have those models, then we can contribute with different engineering tasks, right? From R&D to design, engineering design, to process operations. And this figure, reprinted from my colleague Pantelides, from imagining our colleague working behind the GPROMs, which is now at Siemens, right? So combined with modeling capabilities and also the artificial intelligence techniques, especially statistical machine learning, hopefully we can contribute to improve productivity. That's the expectation. That's the vision of using this methods, really. Now, coming back to our, let's say, PSC strategy at DTU for biomanufacturing, we strongly believe in interdisciplinary research and in close collaboration with the biotech industry, because I think it's important to master the technical details and the challenges of this industry to be able to customize and develop the right modeling and PSC strategy and methods, right? We believe each product and host organism presents a unique challenge, and here what we can use is system sensing approach, and then try to take into account the local constraints. And in this regard, our activities is kind of focused on pretty much on experimentation, data collection. We think it's an important step. And then move on to develop the relevant property process modeling, and based on that to build the house, right? Model-based engineering for different end of application. And indeed, in this sense, we very much agree and aligned with the larger community and our colleagues at Pfizer. The right approach will be, maybe mechanistic modeling, or in combination with mechanistic and AI, a hybrid modeling where you can combine the pros of both worlds. So what I'd like to do with you is to feature some selected case studies, if you will, where we demonstrate the use of this modeling in process scale-up in collaboration with few industry partners. The first case study that I'd like to highlight is using modeling in collaboration with Novozymes, a biotech company. And this picture here is showing the pilot plant at this company. You can see these tanks. They are instrumented, and there is a number of them. And this is what you use in order to test and see what kind of conditions necessary to establish to produce this at scale. And the host organism here is filamentous fungus, and the product is enzyme, which is, for example, used in the bioethanol industry. Process is aerobic, and it's a fat batch, and it takes in the order of days, let's say around a week, just to put it in perspective. So here, what we start with the modeling, and we establish a general mass balance framework around the key process variable, the biomass, the product, the substrate, in this case, glucose, right? And then we ensure the mass balance is there, and in related to that, we take into account also evaporation. Something this is not visible in the lab, like 2 liter or lower scale, but something becomes more relevant when you go at a higher scales, really. In addition to that, we take into account some transport and mixing phenomena, which becomes also, let's say, apparent if you work with filamentous type of fungi, really. So you try to take into account the mass transfer limitation due to the viscosity, among other things. And as the basis for the biokinetic model, we use this, let's say, biochemical stoichiometric reaction, which relates the substrate, oxygen, ammonia, among other things, nutrient consumption to produce cells, and the product of interest, and of course, the off-gas CO2 production. So combine this two, and we can then use the weightable measurements. In that case, it is the weight. This is the feeding profile. And then this is the measurement of viscosity and the mass transfer coefficient. And what you see here is the time scale, scaled naturally, and the y-axis is the concentration, right? And this is for the product. So using this and the parameter estimation, all that, then we can use it as a kind of soft sensor to follow, actually, in our case, the viable biomass fraction, which was, for this particular case, not easy to measure, actually, online, and the substrate profile and the off-gas profile, among other things. So we could use it as a soft sensor. We could also then use this kind of model to study some other phenomena, for example, the effect of evaporation. When you work with a company like Novozymes, which has a production site at the various geographical location, from China to Brazil to States to Denmark here, so you're using aeration, so you're using the air, and here depending on the relative humidity and temperature of the inlet air that you're using and the operating conditions of your system, you could then evaluate what is the impact of the operation. And of this figure here shows the operation. This is for 550 liter capacity pilot scale, this simulation. So in a week, you could lose up to 30 kilograms. This is up to around 6-7% of your mass, total mass. So it can become really significant at certain conditions. So it allows you to study this kind of phenomenon, if you will. Now, once you have a model, then you can see if it can help you with the different scale-up strategies. And here, our PhD student have tested the model and how it can help in the scale-up. You define different agitation speed, different aeration rate, different pressure and headspace, different field conditions for the fat patch. And then you can try to evaluate and see if you can consistently ensure the whole capacity of the reactor is filled. So this is the base case, let's say, DO control strategy, and this is where we use the model-based control in a different flavor. And then what you can see that the model allows you to achieve consistent batch fill. And this is important or relevant because you want to, for the downstream separation, you want to be able to predict how much volume that you need to process in the downstream. So you can do this kind of things. Now I'd like to move on to another challenge, and here just to highlight how different and stark the different, let's say, fermentation technologies could be. So one was it took a week at least or days, and the other one here, just example, is lactic acid bacteria cultivation. This is a study in collaboration with Christian Hansen. So the product itself is lactic acid bacteria, which is a starter culture in the dairy industry. If you want to make yogurt or cheese or among other things, you use this kind of bacteria. Now this is totally different fermentation system, let's say. It's non-aerated, it's batch process. It takes in the order of hours, 6 to 8 hours, you name it. A pH is controlled. And the challenges at the production is the gradients at scale, because when you go to the scale, you go to at least 10 or 300 meter cube kind of fermentation reactor. So you naturally have this gradient. And since this fermentation is sensitive to pH, you want to be able to account for these things. And then also monitoring, we only have two sensors, temperature and pH really. And then we have this ammonia solution as a kind of manipulated variable to control the pH to agreeable conditions here. So this is the challenges. And here we're trying to see if the modeling can help address some of the production scale challenges. So again, we start with a base biokinetic mechanistic models, taking into account the biomass growth in this particular fermentation, which is can be inhibited by the substrate, can be inhibited by the product, which is the lactic acid, and there is a certain optimal range for pH you want to take into account. You can also formulate a weak acid-base equilibrium to be able to, let's say, compute the pH. And then you can collect necessary data to try to, let's say, validate this model. And then once you obtain a satisfactory, let's say, performance, you can move on to the next challenge, which is to try to model the gradient. So here you could use CFD. It's a wonderful tool. It allows you to look at different mixing regime, and then you can move from the full CFD scale to compartmental model, and then you can compare if you can get the mixing time constant at different agitation speed. And then you can match that. And then it allows you to use the compartmental model together with the biokinetic model that you have developed earlier, so that you can now actually predict the pH gradient, which is shown in this figure here, the bottom and the top. And since this is sensitive to this even pH variation, you are able to account it in your model. So having this allows you, and then you can compare between measurement CFD model and compartmental model. And this then allows you to do, monitoring among other things that I will show. But I'll take a step back to try to focus on another thing, which is the monitoring technology and sensor development technology. And there is this concept of free-floating sensor devices that our colleagues have been working with, especially my colleague Chris here, to try to collect data from the production scale reactors real-time really. So you throw this kind of free-floating as opposed to fixed sensor location, and it collects data about the location in the tank, but also pH and oxygen depending on the configuration. And then you can compare this data and analyze it and compare it with the CFP model. And you can see that actually it gives you, let's say, compatible information. So you could characterize now the mixing, let's say, regime by online measurement, if you will, which opens an interesting, let's say, opportunities. Now, back to the model itself. Once you have that, again, you can set up a certain, soft sensor scheme by using the measurements available, which is pH and ammonia addition. And then there is a number of unmeasured variables online. This is the biomass itself, the substrate, and the byproduct lactic acid. And this is basically allows you to real-time monitor it, this unmeasured system variables. Unfortunately, I can't run it now, but I hope you can take my word for it that it allows you to real-time monitor the unmeasured variables, which could be relevant for here. Now, moving on, I'd like to touch base another now fermentation, and this is in collaboration with Deo Pharma. This is the PhD project of Atle. And now here we focus on the product quality challenge. And using mechanistic modeling alone doesn't allow us really to predict this quality attributes of the product reasonably well. So here we have the choice of trying to combine the mass balance mechanistic model with an appropriate, let's say, ML or AI technique to be able to address this challenge. So again, here we focus on full-scale data. So this is a picture of the production scale facility at Leo Pharma. And the product itself here is antibiotic. And also it uses, let's say, a host organisms, which is filamentous in nature. And then it takes in the order of, let's say, days. So it has, again, a different characteristic. So we have sampled at the industrial scale 5 batches. We measure biomass substrate product impurity concentration, and our focus is to model this guy, impurity here. And there is online data collection from the system here. And for this hybrid modeling, we have used a serial configuration of neural net in addition with the mechanistic model here. And what we wanted NL to do is to learn from the available data to estimate the unknown kinetic rates in the system because they are not directly measured. So that was, again, you set up the mechanistic model, and then our strategy to link the mechanistic model with the NL, NN model, neural network model, is the following. So we trained our model to learn this, in this case, the kinetic rate for the related substance formation, okay? And then we constrain it in the mass balances of the mechanistic model. And then we do prior, the tuning and the optimization of this model. And here we show the cross-validation results. In the x-axis is the time, and the y-axis is the related substances here. And what you see here is the related substances, progressively increases, but beyond a certain point, then it comes down. So this is the measurement that you can see in this triangles and the model, this hybrid model combined prediction is in the continuous line. And here you can see the parity plot between the prediction of this byproduct of this impurity versus the measurement. And the R-square for the relative substance is sufficiently well. In addition to that, we can also predict some other relevant process variables for monitoring, such as the carbon dioxide evolution rate, oxygen uptake rate, and pH, among other things. And having this kind of models then allows you to use modeling to take into account not only the titer for the API, the main product, but also the formation of the impurities. And in this case, then we can try to study and simulate different, let's say, operation conditions, hopefully, which is shown here. Right, so these are 3 different use cases focused on fermentation, just to highlight that each fermentation has widely different, challenges and technical details to consider. But naturally, in biomanufacturing, you also have a number of downstream separation for the product capture, recovery, and purification thing. And then modeling can also be used there. Here, just a few other examples from our, let's say, group. This is about a modeling in the dairy industry, about the industrial scale meat drying process with this kind of industrial scale dryers. There's the pharmaceutical crystallization. You could do a lot of also modeling and application of for scale up, among other things. And nature, this is an example from the, let's say, monoclonal antibody production, where there is a number of separation steps that you need to consider in a successful, let's say, scale. So zooming out from this particular, let's say, examples to, let's say, broader challenges, there is a number of other challenges that in biomanufacturing, and if we are to realize this promising potential of biology to produce a wide variety of molecules, right? Then there's a number of things we need to consider. Capacity expansion decision from capacity expansion decision to training of the brains, really, with the necessary skills, right? The PhD, the next PhD professionals, to digitalization, and also the silo approach and the data from discovery of the molecule to scale up, and also the sustainability aspects. I'd like to touch down a little bit. So we have seen in our previous, plenary speaker from Pfizer about the, the fascinating landscape of the pharmaceutical supply. This is an example from Novo Nordisk, which we're trying to collaborate with our PhD student Simon. It's not just one unit, primary API production, whether you're using fermentation or not, and downstream separation, but there's also the secondary manufacturing. There is also the raw material, supply. There is your sales, distribution, and marketing. And there is, if you have a larger company with multi-site production and many, let's say, a pipeline to product to manufacture, the decision related to the future capacity, is, do you need capacity, and if you how much you need, if you need, and how you should increase this. These are really very fascinating and complex landscape, and there is a lot of interdisciplinary contribution to this complex problem, from business analysts to prediction of the future demand for a certain product, among many other things, right? You need to factor this, and so and PSC has developed a number of let's say programming techniques to be able to help here, and we are exploring how we can deploy this kind of techniques here. But I think more can be explored here. Related to the capacity, we also need to consider, do we have the sufficient manpower, right, trained with the necessary and relevant skills here? And I had the pleasure here to coordinate what we call ITM, what we call a PhD training program, together with my colleagues from Europe, different PhD groups from Imperial, from Aachen, to experimental group from Strathclyde and Lyon, in collaboration with different life science industries such as Unilever and Bayer, to try to define a training project, really, from experimentation to modeling and optimization and monitoring and control, so that the candidates, are exposed to the technical challenges of this life science industry, and then help them in this way customize or develop the necessary PhD tools for this field. We think this kind of training it will be relevant, and I think not only in Europe, we would probably need to expand this across the globe if we need to supply the necessary brain power to achieve this. Now, coming back to the digitalization, there's wonderful aspects touch upon, but one thing I want to highlight here is actually for some fermentation, monitoring of the biomass, especially in highly viscous and final testing, is not still very doable. But beyond that, actually, what is still very challenging is the protein itself. It's highly complicated, actually, to measure it, and it takes really quite sophisticated protocol to do it in the lab. So you can't really do that online. This really becomes apparent when we did the survey with our stakeholders from life sciences industry back in 2020 about digitalization and bringing it to the life science industry. So one of the challenges is the level of instrumentation. In some units, for example, in fermentation, you may relatively have quite some high data collection, but in some other units, especially in downstream separation, you don't really have that. And especially if you look at the therapeutic production and biopharma, actually this is one of the key challenges in terms of quality assurance and quality control to move to continuous pharma. It still takes days or sometimes weeks to try to evaluate the quality attribute of your, let's say, intermediate or API product. So there is still a number of challenges to bring digitalization to the industry. Among the online measurement technology, there's also the necessary infrastructure, necessary technical personnel, the organization readiness, among other aspects, really. Related to this digitalization and the big data concerns, what we still see a challenge is the silo approach. As we touch upon it, there is significant amount of data generated at the molecular discovery and strain optimization, not least in terms of the omics technology which is used there. But we rarely really use this information here in this molecular discovery stage when we move to the process scale up and development stage. So you have this strain, and then you put it in shake flies or pilot to determine the kinetics and etc. But you do your own experimentation to try to figure out what should be the recipe for scaling up this. So we think there is quite some challenge here to try to bridge this this silo approach, and explore whether this data in the molecule collected the molecular discovery state can be useful when moving to establish a scale-up process. Another thing is sustainability. Quite recently, there was this climate summit change conference sponsored by UN in Egypt, COP27. And a number of pharma companies, they come together with the vision to try to help decarbonize the supply chain of the healthcare. And as it turns out, the healthcare sector generates around 4 to 5% of total global emission. A majority of this is actually early stage processes, such as the raw material extraction and the processing. So in this regards, I think PAC community can play also increasing role in trying to help and bring the carbon footprint of the sector from potential contribution to process efficiency to, let's say, to try to bring in more renewable utilities such as electricity. And with the carbon capture and storage facilities, among other things. So there is an exciting set of challenges still ahead to address in biomanufacturing. So to conclude, there is quite some excitement and euphoria to use the biology as a formidable production platform on its own, really. But often what we see is process is underestimated in this landscape. The PSC community I think has a role to play here. With our demonstrated competences and skill set in other sectors. But one of the unique challenges in this field is each product process is different and unique, and here mastering the technical details of these challenges becomes the key, really, as a community to help the industry. So in that sense, I think data and experimentation is really important. As a PhD, I don't think we can use our tools before the fact, ab initio. We need to work with the experimentation quite closely. And also we need to work together and early on as a team with this at the scale-up effort to try to help contribute to these processes. I think AI has a role to play here in combination of mechanistic model. Our preference is to use as much as possible knowledge and to deploy the AI when we can. To fill the gap in our knowledge here. Again, another aspect with the PAC, system thinking is we develop a lot of models either for scale up or control or monitoring. But one aspect of it is really to maintain and consider how we're going to reuse it during the life cycle of the process. It becomes also an important, let's say, challenge. With that, I'd like to now, thanks to many of past and present PhD and postdoc colleagues, too many to name, who contribute to exploring this landscape of challenges in biomanufacturing. Also, thanks to our industrial partners and the funding agency, which allows us to work closer to the technical challenges that they are dealing with when they go through different scale-up challenges. And then with that, I'd like to take the opportunity to highlight some of the events, such as our summer school on uncertain sensitivity analysis, and my colleague Sayed's organization of quantum computing workshop in collaboration with AICHE, SBE Society in May next year. So with that, I'd like to thank your attention, and we'll be happy to have any question and for discussions.
Thank you, Professor Shin. Very have a minute, some time for a few questions.
Iftikhar Karimi, Gurkan, nice talk. I had a question on slide 31, where you're showing a profile. It seemed to me that there is a lag, which if you were to, there is a lag in terms of development of profile, right? So I was wondering if you had really tried to model that lag rather than trying to fit a curve all the way through. See, yeah, there is a lag. So there is some phenomenon,
That's true.
biochemical phenomena that's happening, which is allowing the bugs to train themselves and then start working or something like that. So I don't know whether you had looked into that.
Thank you, Iftikhar. Yeah, that's a very good question. I fully agree with you. Lag phenomena, indeed, as you rightly call it, we observe it quite frequently. Now, this isn't the formation of the impurities, right? So sometimes we see the lag observed in the production of the primary, let's say, product, which is shown here. This is the product, and this is the impurities. So that's a wonderful question, and that's really touch on the motivation of using this kind of the black box machine learning aspect. We don't have sufficient mechanistic information at the metabolic level. We don't know if this impurities is formed during the metabolic conversion, of this, let's say, pathway, or if it is produced in the broth of the fermentation due to chemical modification. So this part is unknown. And in terms of, we could ideally go back to and work together with our colleagues from, the metabolic engineering to try to set up and identify and validate a detailed metabolic network model and better understand and get their insight into it. That will definitely be useful to get more insight and mechanistic understanding. But in terms of the, let's say, the engineering application, that was our motivation to couple it with machine learning black box model. Now, related to your question, can we improve the prediction quality? If you want to, yes, we can. But actually, we're not interested in predicting the delay here. What we are interested in, actually, our main thing is to predicting the end concentration here. So when is the time that this impurities start to come down? I don't know if it helps. So this is our motivation here, not to get the start of the formation of this, but basically the end titer concentration. I hope it helps.
Yeah, thank you very much. Yeah.
Sure.
Raj?
Thanks, Gurkan. Thank you for a very interesting presentation. So my question was, again, in the context of the introduction, where you said the road to pharma manufacturing is littered with a lot of technologies which just failed, which never made it. So is there something fundamentally new that we are doing now, which is different from the old cases where failure was the outcome?
This is wonderful question. I want to go back here, actually. Yeah, let me go back to this slide there. Wonderful question. One thing that hopefully we are doing fundamentally different is to become more aware of the challenges and to anticipate in advance the likely process scale-up challenges. We have this silo approach, as I mentioned, that the team that is even if you go today to biotech companies, right, the established time, you still have this silo approach where you have this team which is primarily focused on discovering the molecule and engineering the host to try to express it as a high titer, right, yield. So one thing that we based on these failures and hopefully we are now trying to bring this awareness that we need to come and work closer together as a discipline which is related to just scale up, and to come together and try to see if we can bring this data and experiences from this side, the molecule side and the process side, and hopefully from there, if we can, let's say, anticipate in advance the process scale-up challenges, and if we can give this feedback to our colleagues, maybe to use another host organisms, among other things. So that's a wonderful question, and I would say we are still working on raising this awareness. We are still working on establishing bridge to try to work together between the two communities. So it's not just sufficient to discover an interesting molecule. You also need to anticipate the challenges there. And hopefully with this big data analytics, it's not yet there. We can bring this, experiences together and try to see if we can do it smarter.
Great. So sort of corollary to the same thing. So if I paraphrase what you're saying, it's not purely a technical challenge. It's also the way the companies are internally organized and how they pass off information from one, let's say, the team which operates the lab to the guys who eventually do the manufacturing and so on. So perhaps this is also a structure of the company which leads to some of these issues. Is that a correct interpretation?
Yeah, thank you. I mean, let me put it this way. I mean, maybe this is the reason, but definitely it is the case in the current landscape. In the current landscape, that's really the case. We need to learn to work closer together. But I don't know if this is, how can I say that? Maybe it's also bottom up, right? Maybe in academia, we have trained to think really silo. Like if I'm a PhD engineer, I only work on, what I'm good at. And if a colleague is in the biology department, they only focus on what is they are good at, like, to engineer the organism and discover that. Maybe this is not sufficient to make the successful story. Maybe it's a bottom up, and maybe the current organization of the corporate is a product of this kind of like, I don't know, thinking in isolation or in segregation. So maybe it's both ways, yeah. But definitely that's what I believe, and this excitement about big data is and it's about computing things. Maybe it will help us to converge and come closer. But I agree with your phrasing. This is definitely the current picture.
Thank you.
Thank you very much. And yeah, that's all we have time for the questions. So let's thank the speaker once again.